Geraint, and thanks very much for organising this seminar today. So my lab's interested in the evolution of plant form, and particularly we want to know about the genetic basis of body plan evolution in plants. Oops, that's also not advancing on my own screen now. Here we go. Great, so I thought it would be really fun to just illustrate what I mean by body plan evolution and why it's important by showing you this cartoon overview of animal and plant evolution. So if you read this, from the bottom is older times, up to the top is the current day. And it starts in the Precambrian era over 500 million years ago and moves up in time through to the modern day. So multicellular animals originated from choanoflagellates, which are single cells, and they underwent a Cambrian radiation in body plan in the sea, which is marked by the arrow on the left-hand side. And animal body plans arise during embryogenesis, and body plan diversity reflects changes in HOX gene activity during embryonic patterning. And this patterning sets the number of body segments and their identity, for example, as thorax or abdomen or limb-bearing segments. So if you have a look at some of the little critters on the bottom left-hand side next to that white arrow, so maybe you'll be able to see that they have a different number of segments, and some of them have legs on them and others of them don't. So that's what I mean by body plan diversity. And animals diversified following the origin of major phyla in the Cambrian era, and they colonized land from the sea many times. So you can see moving up through the time in the cartoon, there's creatures starting to appear on land, and gradually they get more diverse through time. In contrast, land plants arose just once from freshwater algal relatives that were already multicellular. And the body plan of today's most dominant vascular plant group reflects diversity in the pattern of development of shoots, roots, and leaves, and these organ systems develop post-embryonically. The major organ systems of vascular plants originated in the Devonian era, around 420 million years ago, and that's where the white arrow is on the right-hand side of the diagram. The Devonian body plan radiation paved the way to a tenfold increase in plant species numbers, so it massively impacted on the biosphere, the geosphere, and the atmosphere. And my lab wishes to identify genes enabling the Devonian radiation of plant body plans, and we also wish to identify genes enabling body plan innovations as plants first started to emerge on land between 515 and 470 million years ago. So next slide, please, Geraint. The closest starting point for body plan radiation in plants probably looked something like this moss plant, with a single stem that's highlighted in the white oval box, capped in a reproductive sporangium. So the swollen bit at the top of that stem is the reproductive sporangium. Please note that this is the diploid stage of the life cycle, and the plant highlighted is called the sporophyte. The green tissue at the base of the photo is the haploid gametophyte, and this provides food for the sporophyte. And I'm going to be referring to both of these life cycle stages in my talk, sometimes switching between the two, so please keep these in your mind. Next slide, please, Geraint. In fact, there's some uncertainty about what the earliest land plants look like. So phylogenetic analyses, such as this one that you can see in the slide, show a divergence from the last common ancestor of the land plants into two major groups, the bryophytes on the left and the vascular plants on the right. So the last common ancestor of the land plants would be right at the base of this tree, where you have the first divergence moving up from the bottom. And along with liverworts and hornworts, mosses form part of the bryophyte group, and bryophytes all have a single sporophytic stem and lack vasculature. In contrast, vascular plants include lycophytes, manilophytes, and angiosperms on the right-hand side of the tree, and these will have branched and indeterminate vegetative shoot systems that you can see illustrated in the little schematic diagrams at the top of the tree, and these arise prior to reproductive development. So there's a really wide morphological disparity between the sort of single stem with a bobble on top that you have in the bryophytes on the left and the indeterminate shoots that you have in the vascular plants on the right-hand side. However, amongst the earliest land plant fossils, there are some, like the ones that you can see along the right-hand side of the tree, in particular Partita theca, that lacks vasculature but branches just once or twice, having features that are intermediate between living bryophytes and vascular plants. The earliest vascular plant fossils, like Cooksonia, have a similar form to Partita theca, but they're a bit bigger and they're more branchy than Partita theca. So if we compare the features of living bryophytes to these fossils, we can gain insight into the features of the earliest land plants. So next slide, please, Geraint. I wanted to show you some photos of what these early land plant fossils look like, and I've included a moss for reference on the left. And then the first fossil moving right across the slide is a tiny fossil similar to Partita theca from Clee Hill in the Welsh borders. And these are just a few millimeters tall, so this is an SEM image, and the stem's really very narrow, lacking vasculature, but the shoot system or the stem has branches that terminate in sporangium formation, and those are the sort of swollen structures that you can see at the top of the shoot there, or the stem there. Cooksonia fossils, moving over the slide, again to the right, have vasculature. So this is what the earliest vascular plants look like. And they're up to about 30 centimeters tall, again with branches terminating in sporangium formation. So those swollen structures at the tips are again the sporangia. And the stems were really narrow and probably lacked photosynthetic tissue. Later vascular plants, such as the aneurophyte on the right-hand side of the slide, had larger shoot systems with sterile branches that lack sporangia at the tips, and the branches have unequal fates. So you can see that there's a large branch up the middle of that shoot, and then branches going off to the left and right of the shoot, and branching again, with each subset of branches getting smaller as the branching happens. These fossils suggest that early steps in land plant evolution involved the origin of branching and vasculature, and later on sporangium development was displaced out of the shoot tips to enable the origin of indeterminate shoot growth and sterile shoot systems that characterize today's vascular plants. So my lab wishes to understand the developmental and genetic basis of these evolutionary transitions. And in today's talk, I'm going to focus on work that we've done looking at one of the characteristics enabling vascular plants to diversify, which was the origin of indeterminacy. So next slide, please, Geraint. Indeterminacy had a single origin in vascular plants, and I've highlighted where that would map on a phylogenetic tree, showing the relationships of living plants with a blue circle on this slide. The indeterminate growth properties of each major vascular plant lineage were inherited from this shared common ancestor, but we can't use fossils to do genetics. So how can we identify genes enabling the origin of indeterminacy? Next slide, please. Most of what we know about the genetic basis of indeterminacy comes from studies in flowering plants, such as Arabidopsis, and indeterminacy arises by the action of multicellular meristems at the shoot tips, which I've illustrated in the schematic in this slide. And these comprise a stem cell zone, which is shown in pink, and a proliferative zone, which is sort of subtending the stem cell zone that's marked in blue on the diagram. And in Arabidopsis, there are two key genetic pathways that regulate indeterminacy in these shoot tips. The clevato-vuschal pathway maintains the size of the stem cell pool at the tip of the shoot as it grows, and that would act in the pink part in the middle of the diagram. The KNOX pathway has a broader role in promoting proliferation and maintaining the balance between indeterminate meristem identity and determinate leaf fate, and that would act in the blue part of the diagram. We're looking at both of these pathways in my lab, but in this talk, I'm going to tell you about how we think KNOX genes have contributed to the evolution of shoot indeterminacy in the vascular plants. Next slide, please, Geraint. Roles for KNOX genes in promoting indeterminacy were first identified by loss of function mutant phenotypes, for instance in the shoot meristemless mutant of Arabidopsis, which entirely lacks a shoot apical meristem. And you can see the phenotype of those mutants in the middle of this slide. So we have a wild-type plant on the left with 2 cotyledons and then about 6 leaves coming from the shoot apical meristem. And in the shoot meristemless mutant, you can see there's just the 2 cotyledons and there's a hole kind of in the middle where the meristem would usually form. So this is the loss of function KNOX phenotype for shoot meristemless, which is one of the KNOX genes in Arabidopsis. And gain of function mutations confer a converse phenotype, shown on the right-hand side of the slide. So for instance, if you overexpress the Arabidopsis KNOX gene brevipedicellus, this confers leaf lobing and compounding onto leaves, and it can also induce ectopic meristem formation on leaves. So these phenotypes show that Arabidopsis KNOX proteins are necessary and sufficient for meristem activity and indeterminacy. Next slide, please, Geraint. The pathways acting downstream of KNOX proteins to promote indeterminacy have been identified in Arabidopsis, and shoot meristemless, or STM, acts to upregulate transcription of the cytokinin biosynthesis gene IPT7. IPT7 expression in the shoot meristemless mutant background can compensate for loss of shoot meristemless function, and KNOX proteins act via IPTs to promote cytokinin production and cell proliferation in Arabidopsis, thereby providing a mechanism for indeterminate growth. Next slide, please, Geraint. Expression analyses in a lycophyte. So if you read this slide back from the tips on the vascular plant side, compare the lycophyte to the angiosperms. The expression analyses in the lycophyte showed that, as in Arabidopsis, the KNOX genes are expressed in the blue part of the shoot tips, which is the proliferative zone of the shoot tips. And working back through the backbone of the tree in this diagram, this finding told us that roles for class 1 KNOX genes in promoting proliferation and indeterminacy are likely to be a conserved or shared feature of the vascular plant group and inherited from a shared common ancestor of vascular plants. So this moves questions about KNOX function and the evolution of indeterminacy out into the bryophyte sister groups of the vascular plants. Next slide, please, Geraint. Meristem organisations change through evolutionary time. So although the vascular plant meristems all have distinct stem cell and proliferative zones, so if you look at the kind of pink part and the blue part in the right-hand side of the tree here, you can see that the sort of overall structure of the vascular plant meristems is broadly similar between the different vascular plant groups. But bryophyte groups on the left-hand side of the diagram don't have a very well-defined meristem in the sporophyte, and there's also a lot of variation in cell proliferation patterns between the bryophyte lineages. So if you have a look at the sporophytes of the liverworts and hornworts, they have a similar overall shape, but these are lengthened by general proliferation or proliferation in what's called an intercalary region, which is marked in blue on this diagram. Mosses are also lengthened by intercalary proliferation next to the liverworts and hornworts, but there's an apical stem cell which is marked in pink that acts transiently to establish the apical basal axis of the moss during embryogenesis. The origin of indeterminacy in vascular plants coincided with the juxtaposition of stem cell zones and more rapidly proliferative zone in the shoot tip. So whereas you've got the pink and the blue separated in the moss, the pink and the blue together in all the vascular plant lineages. And my lab's trying to work on how these stem cell and proliferative functions first arose using reverse genetic approaches. Next slide, please, Geraint. To start to look at how indeterminacy originated, we're using a moss model system, which is Physcomitrium. There are a couple of KNOX gene classes in all land plants, and the ones that regulate indeterminacy in Arabidopsis belong to class 1. There were 3 specific hypotheses that we wanted to test in relation to class 1 KNOX function. The first one was that class 1 KNOX genes are necessary for axis extension in a moss. And secondly, we wanted to test that class 1 KNOX genes are sufficient for axis extension in a moss. And finally, we wanted to test the hypothesis that class 1 KNOX genes act via a conserved downstream pathway in a moss. Next slide, please. There are 3 class 1 KNOX genes in Physcomitrium or model moss, and these are called MKN2, MKN4, and MKN5. Previous work in Mitsuyasu Hasebe's lab engineered loss of function mutants and characterized their phenotypes. MKN2 mutants, which you can see in the slide, have the strongest mutant phenotype, and these have a shorter CETA, which is the stem. So if you follow the dotted lines at the base of the diagram and at the top of the diagram, the dotted lines across the top show the size of the sporangium, and it's fairly similar between the wild type and the mutant lines. And the dotted lines at the bottom would show the equivalent portion of the base of the stem. And then you can see there's a part in the middle that's larger on the photo on the wild type plant than it is with the mutant plant. And the 2 insets on either side show the sort of cell outlines for those portions of the stem or the CETA in the wild type versus the mutant plant. So you can see that there's a shorter stem, but in this previous work, it wasn't really clear whether this short phenotype corresponded to a defect in intercalary meristem proliferation or whether it arose for another reason. So Johan Couder, who did his postdoc in my lab, first grew the mutants made in Mitsuyasu Hasebe's lab, and his phenotypic characterization showed that there's fewer cells in the middle of the stem, and this is what we'd expect if intercalary proliferation is disrupted. We wanted to know whether the mutant phenotype arose at the developmental stage when the intercalary meristem, so the blue part of the plant that I showed you in the previous slide, is activated during development. So, however, sporophyte development's not very well characterized in mosses, so we first staged development on the basis of some morphological markers. Next slide, please, Geraint. We did this by dissecting a lot of developing embryos out of the mother plant and classifying them on the basis of some consistent morphological markers that they had. So starting from the left in stage 1, there's an apical cell at the top and the bottom of this embryo, which elongates the top to bottom axis of the embryo, and that's the marker for stage 1. In stage 2, the apical cells continued to divide, but derivatives were also dividing. In stage 3, the apical cells ceased division, but derivatives continued to divide. In stage 4, where you've got the white line on the right-hand side of the embryo, there's a focus of proliferation that starts, and this is in the intercalary region, which is called the intercalary meristem. So subsequent stages of development are marked by sporangium initiation in stage 5, sporangium swelling in stage 6, and differentiation in stage 7. So stage 4 is the one that we were really interested in, as this is where the intercalary meristem became active. Next slide, please. To determine when the MKN2 mutant phenotype arises, we stage mutant development in comparison to wild-type plants. So you can see the mutants at the bottom there, and Johan pulled out a lot of MKN2 mutants, many more than in the wild type. And we were unable to find stage 4 embryos defined by the active intercalary proliferation in these mutants. So this led us to the conclusion that MKN2 is necessary for intercalary meristem activity. Next slide, please. We next wanted to test whether KNOX gene activity is sufficient for intercalary meristem activity. Next slide, please. And we used a couple of different experimental approaches to test this. So the first used coarse overexpression of MKN2 under a maze ubiquitin promoter. So you can see the construct design at the bottom of the slide. And we used this construct to transform a wild type moss background. So you can see the wild type plants on the left hand side of the slide. So we've got two different mutant lines here, and the overexpression of the MKN2 led to lengthening of the stem. So you can see the wild type stem is sort of short and thick, whereas the overexpressors look a bit long and thin and wobbly relative to the wild type plants. But these plants were pretty much sterile, so Johan was only able to pull out a few sporophytes from each of the overexpressor lines, and it wasn't enough to be able to do a quantitative analysis. Next slide, please. For this reason, we switched to an inducible approach in which Johan fused the glucocorticoid, I can never say this properly, glucocorticoid receptor to MKN2. So we've got the construct design underneath again. So it's MKN2GR, and it's driven by a ubiquitous promoter, and Johan used DEX application to induce MKN2 translocation to the nucleus. So this slide shows results obtained from 3 different transgenic lines with the DEX MKN2 inducible construct or a control construct on the left-hand side, lacking MKN2, but introduced into the MKN2 mutant background. So you can see clearly that plants with inducible MKN2 expression, which are the 2 on the right-hand side of each pair on the right-hand side of the slide. So you've got the mock treated and DEX treated next to each other in each pair of pictures across the slide. And in the DEX treated MKN2 inducible expression lines, the seta or the stem is longer following the DEX induction. In particular, the intercalary region of the sporophytes elongated. And this tells us that MKN2 is sufficient to promote intercalary proliferation and axis extension in the moss. Next slide, please, Geraint. The next hypothesis we wanted to test was whether KNOX genes act via a conserved downstream pathway. And to address this question, we moved to the haploid gametophyte stage of the life cycle. So these are the gametophytes of our model moss, Physcomitrium, and the gametophytes make gametes, and fertilization leads to sporophyte development. So to look at sporophyte development, you have to grow the plants through the gametophyte life cycle stage. And when we were growing our MKN2 overexpressors, which you can see second in from the left on the top, we noticed that the plants were quite a lot smaller than the wild type plants. And the spread of these plants is determined by the growth of a filamentous tissue called the protonema. So underneath the photos at the top, you can see some protonema pulled out from the parent plant, and you can see that the MKN2 overexpressor filaments are shorter than the wild type filaments on the left-hand side. So this phenotype's really similar to the phenotype of wild type plants treated with cytokinin, which you can see second in from the right on the top, and also plants that are genetically engineered to upregulate cytokinin biosynthesis, which you can see on the top right. So this led us to believe that MKN2 overexpressors might have a surfeit of cytokinin. Next slide, please. And to test the hypothesis that there was too much cytokinin in the overexpressor lines, we collaborated with Andrei Novak to measure cytokinin levels in wild type plants and 3 mutant lines. So each of these graphs shows the quantification of a different cytokinin with wild type on the left and then 3 overexpressor lines on the right. And this showed that many different cytokinins are overproduced in the mutants, suggesting that MKN2 promotes cytokinin biosynthesis. Next slide, please. We wanted to know whether MKN2 acts via IPT cytokinin biosynthesis genes, and previous work identified 6 IPT genes in Physcomitrium, but Johan repeated the blast searches and he pulled out 7 genes marked with a blue star on this tree, and 1 gene encoded a truncated protein, so we excluded it from further analyses as we think it encodes a pseudogene. qPCR using our inducible MKN2 line showed that one of these, PPT-IPT3, was upregulated by MKN2. Next slide, please. We wanted to know whether disruption of this gene, IPT3, caused similar phenotypes to disruption of MKN2 function. So if you look at the gene diagram at the bottom, we engineered CRISPR mutants with a range of effects on the IPT3 locus. We've got the wild type IPT3 locus at the top of this diagram, and the position of the sgRNAs that Johan put on is shown by the yellow and red and blue arrows on that diagram. So we got a range of mutants out of that. Milder alleles had deletions, which you can see for the CRISPR1 and CRISPR2 alleles. The effect of the gene structure is shown on the right-hand side. And then there was a stronger allele, CRISPR3, which was a truncation, and the effect of that on the gene structure is shown on the right-hand side again. And the mutants all had a greater area than the wild-type plants. So you can see the overall phenotype along the top. And this is consistent with a role for IPT3 in promoting cytokinin biosynthesis and suppressing the spread of the plants. Next slide, please. In the sporophytes, there was a slight decrease in the elongation of the CETA in the 2 weaker lines, the CRISPR1 and the CRISPR2 lines in the middle of this panel here. But there was a noticeable and statistically significant decrease in the line with the most severe IPT3 disruption, which you can see on the right-hand side of the slide here. So this CRISPR IPT3 mutant has a similar sporophyte phenotype to the MKN2 mutants. Next slide, please. To test whether IPT3 can act in place of MKN2, we used a Moss trick, which is called targeted replacement, where you replace the whole locus in the wild type plant with your mutant locus. So in this piece of work, we used the MKN2 mutant locus, which has a G418 resistance cassette in place of MKN2, and we replaced that with a PPT-IPT3 cDNA. And we wanted to test whether IPT3 can complement the MKN2 mutant phenotype using this targeted replacement strategy. And the outcome of that is shown at the bottom. So we have the MKN2 mutant on the top, on the left, with its short little CETA or stem, and we found that the CETA was longer in each of the targeted replacement lines than it was in the MKN2 mutant. So this told us that IPT3 can restore normal sporophyte development, acting in place of MKN2. Next slide, please. So I've shown you that MKN2 promotes IPT3 expression and cytokinin biosynthesis in Physcomitrium, and this promotes intercalary proliferation in the blue region of the sporophyte axis, shown on the left-hand side of the slide. In Arabidopsis, the class 1 KNOX gene STM promotes IPT7 expression and cytokinin biosynthesis, and again acting in the blue region of the meristem shown on the right-hand side of the slide, and this leads to sustained proliferation of the meristem. Next slide, please. Our data suggested that roles for a KNOX cytokinin interaction predated the origin of vascular plants, because we've shown in very widely divergent bryophytes and vascular plant lineages that they act in a similar way to promote proliferation using similar downstream targets. And this lets us infer that roles for KNOX genes in promoting proliferation are likely to be an ancestral feature of land plants. And I've circled that at the base of the plant tree of life on the diagram here. So we can use the moss model to explore how developmental transitions occurring in the origin of and radiation of vascular plants arose during evolution, and take an exciting peek through a window into the past using these developmental and genetic approaches. Next slide, please. I told you earlier in my talk that there are two pathways that are important regulators of indeterminacy in Arabidopsis, the KNOX pathway and the clavata pathway. My lab's also looking at the evolution of the clavata pathway, and this work so far focused on the gametophyte stage of the life cycle, where we showed that clavata regulates stem cell function. We'd like to move this work to the sporophyte stage of the life cycle to test the hypothesis that clavata was involved in the origin of indeterminacy in vascular plants, perhaps acting with KNOX genes, and we'll use similar approaches to the ones that I've told you about today. Next slide, please. So I'd like to thank Geraint very much for organising this seminar, and thank you also for progressing the slides, and thank you to everyone for listening today. I wouldn't have been able to tell you this story without the work of some fantastic team members and collaborators and the support of my funders. So Johan Couder, who you can see in the photo here, did most of the work on Moss KNOX gene, and various members of my lab are carrying on with the clavata project. So I'd also like you to thank you for your attention, and I'm really happy to take any questions.
Thanks very much, Jill. A second photo of Tom Bennett there on the day as well. I see, different photo this time. So yeah, thank you very much for that. And I once again encourage everyone to ask any questions of Jill as you think of them. So let me kick off. Well, first of all, let me ask. So Physcomitrium, you were saying. What happened to Physcomitrella? Is this something new?
Yeah, it's recently changed its name. So I'm amazed that I managed to get it right. I didn't switch between the different names.
So why was that? Why the name change?
I'm not sure actually.
Okay. So it's noticeable then. So you used the CRISPR mutants, you noticed, to look at the IPT3 phenotypes. Did you also do a complete knockout? So you can do knockouts using homologous recombination in Physcomitrium. So did you also fully knock out the IPT3 as well? Just to see if
we didn't make targeted deletion mutants for that, but the truncation should be a null mutant.
Yeah.
So it would be the same. You can use a range of different approaches to knock gene function out in Physco, which is really nice, and to knock genes in as well.
Okay. And how was using the CRISPR system in the moss? Was that relatively straightforward to do, or do you have some challenges with it?
Well, Johan made it look really easy. We're using Magdalena Betzanilla's, she's made a fantastic set of gateway plasmids for it. So yeah, we're using those, and they worked fairly straightforwardly, and the transformations are just the same as for ordinary plasmids. So yeah, it was quick actually. It was much faster than making targeted deletions. Okay. So that normally takes us about 3 months, but the CRISPR method took about 6 weeks. So it's quite significantly faster.
Okay, I see. So the questions aren't flying in just yet. I don't know if there might be an issue with the chat again that you discovered. So let me continue then. So obviously you talked about the KNOX genes, but then you alluded to looking at the clavata genes as well. So can you give a bit of an overview of what you found so far in that area?
Sure. So we started off the work in a long time ago, maybe 2012. I had my first PhD student pulled out different parts of the clavata pathway, so the Cli peptide encoding genes and clavata receptor kinase encoding genes, of which there are 2 in Physco, and another one that's called RPK2, which has a single gene. We've made mutants for for all of those components. So the clees, there are a lot of them, and we used an amiRNA strategy. And then the receptor genes, we used either a CRISPR or knockout strategy for those. And the mutants were defective in a property that's unique to land plants, which is a capacity for three-dimensional tissue growth. So the mutants are unable to orient their cell division planes in several different directions, which is something that land plants can do, but algae can't do. And the genes were actually specific to the land plants as well. So the gene innovation happened as plants came onto land, and they affect a trait that's unique to the land plants as well. So that was in the gametophyte stage of the life cycle, and sporophytes evolved later than gametophytes. And we've been working on the sort of downstream targets of those with my latest PhD student, Zoe. So she's shown that they're acting to regulate the hormone homeostasis in the apical stem cells at the top of the shoots. And we're hoping to take that to the sporophyte stage of the life cycle fairly soon as well.
Okay. So we got a question from Wendy Moore, very general question, as she states. So any ideas about the origins of the stem cell zone in those early mosses?
Yeah, it would be nice to try and work that out. It's not very clear yet. We can make mosses branch by messing up with their auxin distribution. So if you do that, the plants look pretty like some of those little tiny fossils that I showed you. So there's kind of, it's not direct evidence, but it's some evidence that the apex of the sort of earliest land plants, or the last common ancestor of land plants, probably works something like a moss apex. And I'd like to, that's why one of the reasons I'm carrying on with the moss system, because I'm hoping that it will be able to give us some insights into how the apex would work, and what the genetic regulation of those early land plant stem cell zones might have been.
So yeah, I mean, in your talk you mentioned about the role of cytokine in there, but are you doing any auxin work as well in that area?
The downstream targets of the clavata pathway are auxin related.
Okay.
It looks like. So.
Okay, drawn into it. Okay, so I think we'll leave it there, Jill. Thank you very much. If anyone's got any additional questions for Jill, then please look her up, and her email address is available online. So please ask her anything that comes up. So thanks so much, everyone, for listening. Thank you very much, Jill. Thank you, Laura, as well, for talking. And let you remind you, these talks will be online by tomorrow morning, I should imagine. So please let any of your colleagues know who might be interested in these talks that they can check them out.